Our next speaker is Dr. Chris McGlory. Chris did his PhD with Kevin Tipton in Scotland and his postdoc with Stu Phillips at McMaster, and he's now an assistant professor at Queen's University in the School of Kinesiology and Health Studies. Chris is going to talk about, nutritional attenuation of human skeletal muscle disuse atrophy, mechanisms and future directions.
Okay. Well, thank you very much for the invitation. It's nice to see everybody in person. Today I'm going to speak about nutritional attenuation of muscle disuse atrophy. I'm going to start off by telling you what regulates skeletal muscle mass and size in terms of protein turnover. And then I'm going to speak about the role of amino acids and omega-3 fatty acids in mitigating muscle disuse atrophy. And then I'm going to talk about some future directions and potential clinical applications. So we haven't been as productive as we'd like during COVID, but we've come up with some ideas, and I'd like to share one of those ideas with you all today. So to kind of distill to its essence, the size of muscle is dictated by changes in rates of muscle protein synthesis on the right-hand side there, and rates of muscle protein breakdown. And importantly, both of these processes are going on simultaneously. So whilst you're sitting here now, rates of muscle protein synthesis are occurring, and rates of muscle protein breakdown are also going on. Now, when you consume a meal that is rich in protein or dietary essential amino acids, those essential amino acids are actively transported into skeletal muscle, where they're used as a substrate or to stimulate rates of muscle protein synthesis. And we know that we have an intracellular concentration of amino acids that also does the same thing. Oops, sorry. Now, whilst protein synthesis is going on, we know that muscle protein breakdown is also occurring. So those proteins can be broken down, the constituent amino acids liberated and recycled for the purposes of synthesis, or they can leave the muscle altogether and, say, in the case of alanine, be used as a gluconeogenic product. Now, the important point here is that it's the relative change to synthesis to breakdown that will dictate the size of human muscle. So when you wake up in a rested, fasted state, your rates of breakdown are actually slightly higher than your rates of synthesis. So on a cellular level, you're slowly losing muscle. But when you consume that protein breakfast or that dietary protein, those essential amino acids result in the stimulation of muscle protein synthesis, and via insulin, the slight suppression in rates of protein breakdown that will put you into a positive state of muscle protein balance. And it's the difference between the fed state gains or positive states of balance versus the negative losses that will keep your muscle at that homeostatic level. Now, when you perform resistance exercise or loaded contractions, there's an increase in rates of synthesis, but there's also a slight increase in rates of breakdown, so you're not in a positive state of protein balance. But when you combine resistance exercise with amino acid feeding or essential amino acid feeding, what you see is a potentiated effect or an additive effect, and this is why when you perform loaded contractions or resistance exercise coupled with amino acid feeding over time, you'll see the gradual increase in skeletal muscle size. So whilst resistance exercise has that sensitizing effect towards the effects of dietary protein, periods of disuse or immobilization have the absolute opposite effect. And what they do is that they lead to a decline in fed and fasted rates of muscle protein synthesis that over time will result in a change in muscle size. So here, this is just an example of what we'd see with a fracture or a cast, and you can see around about a 5% decline in skeletal muscle size with around 2 weeks of immobilization, which is generally consistent, but we also see a wide variety or variation in that, particularly in humans. But the important thing I want to stress here is that it's not necessarily those quite abrupt periods of inactivity that can have a negative impact on skeletal muscle protein metabolism. We know that periods of relative inactivity can also have a negative effect. So this is some work that I did as a postdoc with Stu Phillips, where we looked at the effect of daily step reduction on rates of protein synthesis in pre-diabetic older adults. And it's kind of prescient data considering that this was done in 2017. And then we now have the quarantine periods with COVID where we go through those kind of restricted periods of physical activity for 2 weeks. And what you can see here in this study, what we did was that we took healthy or pre-diabetic older adults, and we reduced their step count to below 1,000 steps per day. So it's around 900 steps per day. And we did that because at the time, that was what we knew was around the number of steps that somebody would take if they were admitted to hospital, generally with influenza or something like that. And then we brought them back up to their normal daily step count. And you can see there's quite a variability here in terms of the daily step count of these older adults. But what we found was that those rates of protein synthesis, now this is myofibrillar protein synthesis, the contractile elements of skeletal muscle in older women and men, was significantly reduced, and it was not recovered following return to basal levels of activity. And we also measured indices of insulin resistance in terms of the HOMA-IR. That went up, so insulin resistance went up, didn't come back down at recovery, and insulin sensitivity also went down and failed to fully recover, although it was on the way back. So what hopefully this kind of gets across here is that it's not necessarily those really abrupt periods of inactivity that you associate with bed rest or cast immobilization that is really having a negative effect here. You can see these rather more subtle but pernicious effects of physical inactivity on muscle protein metabolism. And what we also did was that we used we went through a microarray approach to look at some of the genes that were being regulated here or being changed in response to this period of inactivity, and consistent with a lot of reports, we found that there was a downregulation of predominantly mitochondrial-related genes, which is consistent, again, with a lot of a growing body of evidence suggesting that mitochondrial health and function is implicated also in good quality muscle. Now, if we take a step back and think about, well, why is this important? Well, we know that as we begin to age, and after the age of 50, and even from a strength perspective at around 35 to 40 years of age, we start to lose muscle mass and lose muscle strength. And if you were to superimpose these periods of inactivity on that decline, which is commonly referred to as sarcopenia in aging, if we were to punctuate that natural decline with those failed recovery periods, which is oftentimes what we see in older adults, what would happen is that you would breach a disability threshold earlier in life than you otherwise would do. So it's these constant insults of inactivity that you see and failed recovery that would then precipitate that early breach of that disability threshold. So what we're really looking to do in a lot of my research and a lot of the training I've done has been focused on trying to find interventions that would shift that curve back up towards that normal natural decline and even beyond that. Now, I will preface this by saying, you can't out-nutrition physical inactivity, so to speak. You really want to promote activity or exercise, but there are many cases, particularly when people are very sick, that they can't actually perform loaded contractions or exercise. So nutrition appears to be, or at least is one of the frontline remedies there to try to achieve this. Now, at the molecular level, this is kind of, and I appreciate that this is an area that is being advanced month by month here, and this is just a kind of very basic overview of the molecular mechanisms that regulate both protein synthesis and proteolysis, and both processes are very important in regulating muscle size. But in my lab, what we really do is try to focus on the protein synthesis side of things, although I'm aware there's going to be some great presentations on the proteolysis and posters later on in the conference. But from our perspective, what we really want to do is to focus on that protein synthesis side of things, specifically looking at treatments that may target mTORC1 and associated cellular molecules downstream of mTORC1, such as P70S6K1. Now, a lot of work that has been done in cell cultures and human work from Dan Moore's lab and also Andy Philp and others has shown that if you feed essential amino acids or amino acids, what can happen is that mTOR will associate with the lysosome, where it can interact with its coactivator Reb in its GTP-bound state, leading to the downstream phosphorylation of P70S6K1 and 389. So this is kind of one of the mechanisms that we believe by which amino acids or essential amino acids stimulate rates of protein synthesis. You consume those amino acids, they target mTOR, they lead to an increase in association with the lysosome and then an upregulation of protein synthesis and binding of the ribosome to the mRNA. So it would be kind of like a logical extension here to think that if you were to increase essential amino acid or amino acid intake, that you may be able to potentiate those MPS responses during inactivity and then try to protect against those declines in muscle FSR rates and also muscle size. And there's a lot of work out there on this, but I've just picked a couple of studies here that I think kind of illustrate this quite well. And this is work from Tanya Holloway, who was a postdoc in Stu Phillips' lab at the time. And she did a really good study, a well-controlled study here in humans, where she took 20 healthy young men who were consuming around 1 gram per kg of protein. And they went through a 7-day immobilization period, but they had a lead-in and throughout the immobilization period, one of 2 interventions. One intervention was a placebo, and the other one was an essential amino acid mixture that actually had a non-essential amino acids as well. But if you look, leucine there is one of the key agonists of mTOR. That was in the intervention drink there, and they had a 7-day lead-in followed by a 14-day or 7-day immobilization period. So there's a 7-day immobilization period there, and what Tanya showed was that with consumption of those essential amino acids or that amino acid drink, that are protected against those declines in quadriceps cross-sectional area. So importantly, the high-dose essential amino acids appeared to increase, or we thought that it increased FSR rates, leading to that protection against the decline in quadriceps cross-sectional area, and this was measured via MRI. But importantly, what Tanya showed was that this didn't really have an effect on strength. So again, this is something that is quite consistent in the literature. It's a little bit mixed. It's generally consistent that you really can't certainly out-nutrition declines in strength, but at least we saw a kind of decline there in cross-sectional area of muscle size. Sarah Oikawa moved this work on a little bit and looked at the effect of high-dose essential amino acids in the form of whey protein at mitigating the declines of protein synthesis in response to step reduction during an energy deficit. So here Sarah took all these, this time it was healthy older women and men, put them through the same step reduction protocol that we'd done previously. This was during an energy deficit, but with high-dose whey protein, it appeared that, again, there was a slight protection against the declines not against the declines, but there was enhanced recovery with the essential amino acid ingestion there. So whilst this kind of gives us a positive overview of like, well, maybe then we should just simply start increasing our essential amino acid intake, and then we can protect against these declines and mitigate atrophy, but not all studies have shown the same thing here. And this is, again, excellent work from Luc van Loon's group, where they looked at the effect of just giving leucine alone on mitigating atrophy during 7 days of mobilization in young men. And the key difference here with this study is that leucine was started on the day of the immobilization, or just as immobilization began, and they were in a state of energy balance, and they were having quite high protein intake. So it was about 1.2 kilograms per body mass of protein. So kind of typical of what we would see in this population, but certainly higher than the RDA. And in this study, they didn't see a change in quadriceps cross-sectional area, and here's the individual data, and they certainly didn't see a change in size. So it does appear that there may be some slightly mixed results, but the reasons for this could well be that there are differences in terms of state of energy balance, and also whether you start in with like a prophylactic approach and whether you lead in with that high-dose essential amino acid supplement. So just a quick recap on what I've told you up to now is that the declines in rates of protein synthesis are a major driver of muscle tissue atrophy in humans, and there is also an important role of protein breakdown. Essential amino acid ingestion may protect against the declines with mass, but certainly not with strength. And that ingestion of essential amino acids alone are only partially protected against atrophy. And as, again, I just want to reiterate that it is, exercise and physical activity, and we've even seen superimposed neuromuscular electrical stimulation can have a more profound effect, but really nutrition in the hospital is what we're kind of looking at here. So this kind of led us to think about other nutrients that we could look at that could be used adjunct to essential amino acids that may actually help or potentiate that effect. And one of the first studies that kind of that caught my eye was that was conducted in Laval, where they fed growing steers long-chain polyunsaturated fatty acids, or omega-3 fatty acids. And what they showed was that in these animals that were fed with those polyunsaturated fatty acids, or omega-3s, that there was increased rate of disposal into peripheral tissues, or actually out of the systemic circulation, of amino acids and glucose. So the authors speculated that those omega-3s had been incorporated into the phospholipids of the tissues of these animals that improved the transport of these nutrients across the membrane and uptake into those tissues. But the limitations of these data here is that it's whole body, so we don't have anything related to muscle-specific data. It's also in animals as well. So taking this work forward, Bettina Mittendorfer did one of the landmark papers in the field with omega-3 fatty acids. And what she did was she looked at the effect of long-chain N3 fatty acids in the form of omega-3s on rates of muscle protein synthesis in response to amino acid infusion in middle-aged and older adults. And I'm just going to talk you through these data here. So on the y-axis, we've got mixed rates of muscle protein synthesis before and after 8 weeks of 5 grams per day of omega-3 fatty acids. And this is in humans. Now, before supplementation, they came in, basal rates of FSR were measured, and then they were hit with the infusion. And as you would expect, I've already told you that essential amino acids stimulate rates of protein synthesis, and we see an increase in rates of FSR, which is what we would expect. After 8 weeks of supplementation, there was a 2-fold increase in the omega-3 content of the membrane. The two key omega-3s I'm talking about here are EPA and DHA. 2-fold increase in the membrane. Rates of FSR were once again assessed, but there was no effect of omega-3 supplementation on basal rates of muscle protein synthesis. But this time when they were hit with the clamp, there was not only an increase, but there was a potentiation compared to before supplementation. So somehow, these omega-3 fatty acids were being incorporated into skeletal muscle membranes, and they were rendering skeletal muscle more anabolically sensitive to the provision of amino acids. And then when they took a look at the molecular level and some of the signaling molecules that I've talked about already, they found that this was associated or linked with increases in both mTOR and its downstream target P70S6K1 on the 389 site. And again, they followed up with another study and shown that there was an increase in mitochondrial gene expression, opposite to what we had seen when you go through a period of inactivity or disuse. So whilst this, when the study was published, there was also a large number of other studies coming out related to omega-3 fatty acids. This one here is from Stuart Gray's group in Scotland, showing that when you feed omega-3 fatty acids to women and men undergoing resistance exercise training, there was an increase, or they potentiated the gains in strength. Graham Holloway's lab had shown that when you feed young men with omega-3 fatty acids, it altered mitochondrial respiration kinetics. And then others from Brazil had also shown that when you feed these omega-3 fatty acids, it seemed to enhance the strength gains with resistance exercise training. So we're starting to see from different labs, from different research groups, that there's a kind of unifying message here that these omega-3 fatty acids seem to be anabolic in nature, but we still really don't know how or why. But one paper that really caught my eye was one that was done in preclinical models that showed when you feed omega-3 fatty acids to these animals when they go through a period of immobilization, it protected against the declines in myosin heavy chain content. So here's the primary data from that study showing that in these animals, when they went through a period of immobilization after consuming dietary fish oil, there was a protection against declines in myosin heavy chain content. And this again was linked to the phosphorylation of P70S6K1. So again, some limitations here. Preclinical model, is this really relevant to the human field? Could we translate this and see whether omega-3s could work in that capacity? Well, at the same time, we were doing our own project where we looked at time course changes in omega-3 lipid profiles with supplementation. And what we showed was that whilst there was an increase here, and again, I appreciate there's no control group, but whilst there was an increase in the EPA and DHA composition of the lipid membrane, this was also linked with an increased expression of total focal adhesion kinase. And total focal adhesion kinase is a mechanically sensitive protein that feeds into the mTORC1 signaling cascade to upregulate protein synthesis. So to try to pull all this together now is what we were thinking, was muscle disuse results in muscle loss, and it also reduces the sensitivity of protein synthesis to amino acids. So that's the primary mechanism here. Not to say that there's not an important role of breakdown, but we believe in humans that one of the most dominant mechanism that is driving this loss is the desensitization of muscle to the provision of amino acids. This is associated with a reduction in mitochondrial gene expression, whereas on the other hand, omega-3s are increasing rates of protein synthesis and enhancing mTORC1 signaling and mitogene expression, so having the opposite effect. So what we thought to do was we teamed up with Graham Holloway at the University of Guelph and Paulo Maiotto, and we wanted to ask the question, is if could omega-3 fatty acid intake restore the sensitivity of skeletal muscle to amino acid provision during immobilization and reverse those effects? And this was done here with Raveen Baniwal and Mike Kamal when I was working with Stu Phillips in his lab, and I'm just going to talk you through those data now. So we had 20 young healthy women, 11 in the omega-3 group, 9 in the control. Very similar in terms of age, body mass, lean body mass, body fat, and strength. They came into the lab. We took a rest and venous blood sample, a muscle biopsy. They began supplementation with 3 grams EPA, 2 grams DHA. That was for a period of 4 weeks, and that was based off our previous time course data. Again, another venous blood sample and a rest and muscle biopsy was obtained. They then went through a period of 2 weeks of single-leg immobilization, serial muscle biopsies, and then MRI for size and DEXA for lean tissue mass were obtained before and after. And then they went through that free living recovery period where once again we took a post-muscle sample and measured changes in muscle size and muscle mass. Here's the data in terms of the compliance and also changes in EPA and DHA composition of the muscle. We can clearly see here that there was a significant increase and a plateau after about 6 weeks in the intervention group and no change in the control. And the money figure here being that there was an actual protection against the declines in peak quadriceps cross-sectional area in that omega-3 group. There was also no statistical change, there was a preservation of leg lean mass measured via DEXA in the omega-3 group as well. And then tying it all together, we looked at changes in integrated rates of muscle protein synthesis, and as we would expect, in both groups there was a decline in rates of FSR, or muscle protein synthesis, but there was a main effect of omega-3s in that rates of muscle protein synthesis in the omega-3 group were consistently higher across all time points. So this would suggest to us, again, feeding back into the previous data from Bettina Mittendorfer and Gordon Smith's work, that it seems that these omega-3 fatty acids are being incorporated into the tissues and having an anabolic effect, but we really don't know why. And we were kind of very lucky enough to team up with Graham and Paula, and Paula was at Guelph, and she'd drive down Highway 6 every single morning, sometimes at 4:30 a.m., collect the tissue, go back to Guelph and do the respiration measures. And what we found was that, or what she found was that in the control group, that there was a significant decline in ADP-stimulated respiration, or oxygen consumption in the control group, but not in the omega-3 group. So it seemed that, again, consistent with the previous work from Graham's lab, that omega-3s were affecting mitochondrial function in this capacity as well. So kind of taking a step back, we really are thinking at this stage, well, how is this working? It seems in some way too good to be true. You see these big protections against issues atrophy, but then again, the other side of it is, you've got different labs showing very similar effects, and kind of reproducible effects as well. So it seems like something is going on, but we don't really know why. We've done some probing analysis around amino acid transport. We don't really think it lies there. We think it may be related to some kind of mTORC1 signaling cascades. We haven't measured protein breakdown, largely because it's very difficult to measure in humans, so we're not really too sure there, but we're not convinced. The natural thing when you think about omega-3 fatty acids is the anti-inflammatory effects, but the important point is in a lot of the studies that have seen these anabolic influences of omega-3s, it occurs in the absence of any change in circulating or intramuscular inflammatory or anti-inflammatory markers. So we don't think the mechanism is there. But we recently published a paper, and this is led by my grad student, Emily Ferguson, where we think there may be something quite nicely tied in here with the mitochondria, and there could be some kind of retrograde signaling mechanisms between the mitochondria and the factors that regulate muscle protein synthesis. And there's been some early work coming out recently around ATF4, and ATF4 is thought to drive atrophy, but it also may actually regulate mitochondrial health and mitochondrial function, and I believe there's a good poster from Dave Hood's group going to be up in the next couple of days that speaks to this effect as well. So we think there may be some kind of crosstalk there, which would make sense, because if you think about the mitochondria producing the majority of the energy in the cell, and muscle protein synthesis is an energy-consuming process, or a highly energy-consuming process, a crosstalk mechanism linking the two would be kind of logical or biologically logical to us, but we still really haven't probed this too much yet. So for the final 2-3 minutes, what I'm going to do is just share with you a proposal. So we were hit like everybody else pretty hard with COVID. Started in 2019, and then we were shut down for 2 years. So we spent some time thinking and writing some grants, and we were very lucky to be able to secure a CHR project grant to try to take this omega-3 work forward, and in the context of bed rest. So this is a bed rest facility that we have at Queen's, where I'm based. And I'm just going to show you the protocol that we're looking to do here, and it's not finalized, but I'd be willing, if anybody would like to talk afterwards, to talk about the protocol or potentially collaborate. We're also looking for trainees to help run this project as well. What we're going to do is we're going to take 60 people, 30 control, 30 intervention, and the end size is due because we're going to stratify by sex. So it's going to be women versus men. They're going to be young. We're going to load them with either control or omega-3 fatty acids plus essential amino acids for a 14-day lead-in, then through a 7-day bed rest protocol, muscle biopsy samples as per our previous work throughout, MRI and DEXA throughout, and our primary outcome is going to be the clinically relevant change in muscle size, and we're also going to measure muscle strength as a secondary outcome. We'll follow up with some EM analysis and immunohistochemistry for fiber type work, mitochondrial respiration measures, which we've just secured in our lab, where Emily's going to start probing a little bit to see whether we can tease out any mechanism between the mitochondria and also mTORC1 signaling mechanisms. But one aspect that we're kind of pretty excited for as well here is that we normally use deuterium or D2O to measure protein synthesis, but one thing that has been kind of an answer, or has eluded a lot of, it's a question that's eluded a lot of researchers is, well, we know that protein synthesis is heavily affected, but what about rates of protein breakdown? It's actually very difficult to measure in humans. But we're going to collaborate with Yatin Bernstein at Liverpool John Moores, where he's developed a dynamic proteome profiling approach that not just enables us to measure the synthesis and breakdown rates of proteins, but also at the individual level. So we'll be able to see which proteins are being synthesized, which ones are being broke down, and if needs be, collate them into bulk fractions so that we can actually see which particular fractions are being regulated. And this may also help us tease out some things in terms of the mechanisms as well. So just the take-home messages, declines in rates of MPS, or muscle protein synthesis, predominantly drive muscle disuse atrophy in humans, but there's a relatively unknown contribution of muscle protein breakdown. Essential amino acids and omega-3 fatty acids attenuate muscle mass losses, but not strength. And omega-3 fatty acid therapy could counteract disuse atrophy during simulated hospitalization, but we are yet to see. And finally, I would just like to thank all the collaborators, and particularly Stu Phillips, where I was trained, Queens, where I share a lab with Brennan Gerd, and we're looking forward to doing some exciting stuff the next few years. Graham and Paula, particularly for their contributions, both theoretically and also to getting the studies over the line. And then the University of Stirling, where I was trained, and I think it'd be remiss of me not to acknowledge my late supervisor, Kev Tipton, who unfortunately passed away, but he put me on the track to a lot of this work, and I'd like to thank him as well. So if anybody has any questions, I'll be happy to answer them, either now or later on as well. So thank you.
Sure, we're going to get lots of questions. Chris, let's see. You're up first.
All right. Hi, Yun Tian from University of Virginia. So I saw that in the omega-3 feeding, there was some profound protection of the mitochondrial respiration in the human samples, right?
Yeah.
I was wondering whether you took a look at any other properties of mitochondria, like are there overall content difference or morphology, dynamics?
So Paula really drove that work. Off the top of my head, I believe there may have been changes in mitochondrial oxphosphate content. I can follow up with you afterwards. I think Graham might be in the audience, or Paula, I'm not too sure. I do think ANT1 might have been altered, but again, I can't give you a definitive answer off the top of my head, but I can certainly have a look and talk to you, but it's in the FASA paper that they published.
Okay, thank you.
Hi Chris, Dan from the Norwegian School of Sports Sciences. Thank you for your nice presentation. So I noticed in the bed rest study you proposed that you were going to compare the males and females.
Yeah.
I wondered if you'd done that in the unilateral limb suspension models that you've previously done, and do you see any differences between genders?
No, so that's a really good question. So we've not made that comparison. One of the reasons it's difficult to make that comparison, particularly with the human work, is it's just extremely expensive. If you start powering studies with a primary outcome where you have gender or sex as an effect, and then at the end of the day they cut you 20% anyway when you get the grant, as we all know. So it makes it difficult to really pursue those questions. But the reason we chose women in the immobilization study was that previous work that had done males versus females in terms of the resistance studies had shown that women had a more potentially greater gains in terms of strength, and there's a lot of evidence to suggest that women can actually convert EPA to DHA far more efficiently than men. So there's some evidence that there's sex-based differences in omega-3 metabolism. So that's why we pursued the work in women. But to answer your question directly, no, we've not done that comparison.
And what about just general sort of size and strength measurements? Do you see any differences in?
We've never done a study in which we've had both in the same group, but I do believe Bettina Mittendorf at least has seen some potential sex differences there. Stu Gray's group detected the sex difference. That's the only study I know that has done women and men that is statistically powered, but again, it wasn't a primary outcome, so how sure we can't really hang our hat on that until we start doing these studies that a priori powered, reported, and then we follow up.
Thank you.
Hi, I'm Bruce Spiegelman. Maybe I'm prejudiced by having interacted a lot with Fred Goldberg and Marco Sandri, but I remember those guys publishing very convincing data in animals, I believe mostly, that disuse induces tremendous induction of attrogenes, E3 ligases against the myofibrillar proteins. And I was under the impression that in experimental animals, that muscle protein breakdown was really activated by disuse. So is this a species difference?
So firstly, I think there are major species-specific differences that have a bearing on this debate that can sometimes confound the issues. So the issue with the rodent work it's not an issue, it's just a limitation, is that oftentimes it's limited to snapshot markers, and that measurements of protein breakdown are actually very rarely even measured.
But Fred was the pioneer in that. I think nobody could do it better than him.
True, but I mean, I'm not, again, entirely familiar with the preps, but a lot of the time it's not, I don't believe it to be to replicate the in vivo measurement or the in vivo biological factors that regulate in humans. There's obviously higher rates of metabolism, higher rates of FSR turnover. They seem to be predisposed to showing increased rates of these atrophy genes compared to humans. But having said all of that, one thing that we've got with the human to kind of balance that out is a lot of our human work is measured in healthy people, largely university students. How our work would translate to a hospital setting where somebody is there with increased sepsis or hyperinflammatory states, where it's quite likely that in that particular setting, breakdown would also be increased. So, you know,
I mean, they also used extreme models like denervation, starvation, fasting. So maybe maybe they're pushing. I don't know, Marco. I saw him here somewhere. I mean, maybe they were pushing the envelope harder than you would in humans.
I also think, well, some of the preps as well divorce it from an intact circulatory system. So I would assume that if you were to do that and remove like the provision of oxygen and nutrients, it's quite likely that the cell's going to start to trigger a death response. But again, I don't think it should be like butting heads on that issue. I think it may be working together to try to find a way to find what really is going on.
Okay, thanks.
Yeah.
Thank you for the presentation. For the omega-3 fatty acid study, did you try to look at an older population? Because you can see that in the recovery part, they both go back to normal, and this is something that we don't often see with the older population. So you see the drop, and then they have more difficulties to recover.
Yeah, that's a good question. So there was a kind of, we were thinking about what we should do here from an ethical point of view. So if we're going to be putting older people into bed rest, prolonged periods of bed rest, to make it clinically relevant from a time frame, it needs to be 10 days. You know, so the average hospital stay for an older person is 10 days. So to put an older person, particularly an older lady, into bed for 10 days without strong evidence that we think they're going to recover. I'm not too sure from an ethical perspective that was right. So we're going to first do it in younger folks, and then if it does come out, we'll translate it into that older setting.
We actually just finished a study doing 14 days of bed rest in older adults with the 6 degrees head down. We can discuss it because we studied muscle protein breakdown and synthesis. So maybe we'll discuss after
That'd be great.
the presentation.
Yeah.
Thank you for a nice talk. So one of the events that also occurs with the immobilization is the loss of neuromuscular junction integrity. I can't remember how quickly that occurs, but how much do you think that contributes to this reduction in protein synthesis that you see just because the muscles are not stimulated as effectively?
Yeah, I mean, that's not really my area of expertise, but I've read a fair bit of work that has come out, I think it's in Scandinavia on this, and it seems like it's having a profound effect. I'm not too sure exactly of the mechanisms, and it seems to be some kind of like denervation of the fiber. But I think one question that I'm kind of interested in is what is the time course of these changes? So is it denervation, mitochondrial dysfunction, then we see changes in mass? Is it changes in mass here feeding back in and then feeding upstream? And I think this is an area that is quite exciting because are we having this domino effect of one hitting the other, hitting the other, hitting the other, and then we're looking here when actually we should be intervening up here. So it's kind of trying to find a way to establish that time course change. But I think a lot of the time with that, looking at the precision of the measurements that we use, we may get false positives in terms of using very precise measurements for one area, seeing that go up and not as precise for the others. And then the adequately powered human studies for that type of work, I think, is critically important.
Very nice. Thank you very much.
One more.
Hey Chris, great presentation. I have a couple of questions. So do you think these findings are exclusive to supplementation with omega-3? Or I'm just thinking of like whole food, like consuming like fish in the diet, do you think you'd see similar effects?
No, I think that for my approach is always a food-first approach if we can. I think that the quality of food in hospitals is pretty poor. I think that's consistent wherever you are. I think the proof of concept way is firstly to go in with these supplements to establish that proof of concept, and then if we can start to convince people to bring it into from a food-first approach. But again, if you think about people who are in hospital who are pretty sick, trying to consume that amount of protein in a food source, or people who don't like fish or consuming that amount of EPA and DHA in a food source can be quite problematic. So I think in some instances supplementation might actually be appropriate, but I would always say first approach is food.
Great. Second question. So I'm thinking about like sports injuries. Do you ever, with these immobilization, do something like a damaging exercise protocol, something that, you know, say someone tears a muscle or something and then would be immobilized? Do you ever in your models consider doing something like that, where you kind of provide damage first?
We haven't, but I think Ben Wall's done a study recently that just came out where they did damage and exercise prior to disuse, and I actually can't remember what the findings were, but I just simply remember them doing that work.
Okay, great, thank you.
Thank you.
So Chris, why don't you combine your omega-3s with some of Val's lithium chloride there? You'd have a polypill against muscle disuse atrophy.
Polypill, yeah, could do.
Huh? It's a good idea, right?
You could fund it. Take it from your answer, could you?
Excellent. Thank you very much, Chris, and thanks to the speakers this morning.